Okay, so here I'm going to talk about the major histocompatibility complex, which is also known as the HLA, or human leukocyte antigen in humans. And the MHC complex histocompatibility was actually identified during transplant rejections, and it was the major cause of acute transplant rejections when transplants were started. So I want to just go over the development of the major histocompatibility molecule. So what's important to understand about this is it comes from your germline. So this was given to you from your mother and father. You have MHC alleles, and an allele is simply just a gene. And there are many of them that you inherit from your mother, and then you inherit another set from your father. So there are two different types of Mhc class alleles. We have Mhc Class 1, and this is going to be a gene that activates CD8 T cells. And then we're going to also have a different set of alleles that are Mhc Class 2, and these activate CD4 T cells. Now, both of them are going to have to go through the process of transcription which is DNA, of course, to RNA, and then translation, which is RNA, messenger RNA, to protein. So they both do this. And then your class 1 is going to generate what's called an alpha polypeptide. And your class 2 is going to generate both an alpha and beta polypeptides. And a peptide is simply a sequence of amino acids. And of course, your genes are a sequence of nucleic acid. So your alpha polypeptide for your Mhc class 1 will join with another protein called the beta 2 microglobulin. And this will form the MHC molecule. And then the alpha beta for your class 2 will also form an MHC molecule. Key is MHC1 activates your CD8s, MHC2 activates your CD4s. Now let's talk about the Mhc gene itself. So the loci, or where in your genome the Mhc or HLA allele genes are found on chromosome 6. And just sort of a nomenclature is that if you have human leukocyte antigen plus 2 letters, such as DP or DQ or DR, this is going to be MHC or HLA class 2 and activate CD4. And if you have HLA plus 1 letter, A, B, or C, this is going to be MHC class 1 and activate CD8 T cells. So you can see here that we have DP, DQ, and DR, which all have alpha beta because Mhc class 2 polypeptides are alpha beta. And then we have A, B, and C, which all have just alpha because the polypeptides for class 1 are alpha. Now there is a few other genes that are part of this complex, this gene loci, and we have our LMP and TAP. Now LMP is going to stand for large molecular weight proteasome. Basically, it is a proteasome which chops up proteins into peptides that are able to fit into MHC molecules better. And then we have our TAP, or TAP, which is a transporter associated with antigen processing. And so just so for a little bit of reference to cell biology, under conditions of normal, and then you have infected. So in normal, you will have self-proteins, and these will be fed through a proteasome. And these will make self-peptides. And then in infected, you will have, for example, a virus protein, and this will be fed through an LMP proteasome. And this will generate virus peptides. So the LMP is there to make the proteasome a different size so that it generates peptides that can bind more effectively to the MHC molecules. Self-peptides are always made. You're always breaking down proteins. They're always going through a proteasome. And they will get loaded onto MHC molecules. But when you're infected, you want to make sure more of the virus proteins get processed and presented on MHC molecules. So that's why you have the large molecular weight proteasome that's encoded within the HLA gene. Now, the next thing I want to talk about is MHC polymorphisms. So if you look at MHC1 alleles, remember we had our A, B, and C. Our A has about 3,000 known haplotypes, or 3,000 known genes associated with A. B has about 8,000, and C has about 2,500 at the population level. Now, Mhc Class 2, we have our DP, we have our DQ, and we have our DR. So DP has approximately 500, DQ has about 800, and DR has about 1,750. And so, of course, our MHC Class 1s are more diverse than our MHC Class 2s, but they still have a lot of diversity within the MHC loci. Now here I'm showing a father by a mother. So you see the father from his paternal got red MHC loci and purple from his mother. And the mother in this case got a green from her father and then a orange from her mother. So if you look at the offspring, they all look different. So you have offspring 1, 2, and 3, and they all have different MHC loci, which Mhc alleles passed on from their parents. And what this means is that because MHC is codominant, if you have offspring 1, on their surface, for example, they're going to have both green and purple Mhc alleles on every nucleated cell. And so this means if they had to get a tissue transplant, if they got a tissue from any of their siblings, it would be rejected because they're not a perfect match. And so MHC came from transplants, the compatibility, how compatible you are. Your tissue being donated to your recipient, how compatible are the donor and the recipient for Mhc alleles? And so this is very important for transplant and transplant rejection. Now let's next talk about the structures of your MHC molecules. So talk about just some general things. MHC is always going to be membrane bound. It's not like your antibodies that can be secreted. And Mhc Class 1 is going to activate CD8 T cells. That is never going to change. And Mhc Class 1 is expressed by all nucleated cells. So the Mhc Class 1 molecule structure is going to look like this. You have a 1 transmembrane domain. You have your alpha 3. Draw this a little bit better. Transmembrane domain. You have an alpha 3. Remember, Mhc Class 1 has alpha only. You have your alpha 2 and your alpha 1 that make up the antigen binding site. And you have your beta 2 microglobulin. So this is basically the structure. And then the peptide would fit into the peptide binding groove. And so this Mhc Class 1 molecule will be found on a macrophage. It will be found on a hepatocyte, a liver cell. It will be found on another T cell, but it will not be found on a red blood cell. Red blood cell is not nucleated. It cannot express Mhc Class 1. Now let's look at Mhc Class 2. Mhc Class 2 has some of the same restrictions. It's going to be always membrane bound. This time it's going to activate CD8 T cells. CD4 T cells. And it's only APC expresses this. And the APCs are macrophages, also monocytes, dendritic cells, and B cells. And so the Mhc Class 2 is a little bit different. Mhc Class 2 has 2 transmembrane domains. And remember, it has beta and alpha chains. So on one side, it's going to have the beta 2. On the other side, it's going to have the alpha 2. And then it's going to have the beta 1. And it's going to have the alpha 1. And these are going to make up the peptide binding sites. Now, this Mhc Class 2 molecule will be expressed by your macrophage. It will not be expressed by a hepatocyte. It will not be expressed by a T cell, and it will not be expressed by a red blood cell. So only an APC, in this case macrophage, can express. All right, so another concept to talk about is restriction binding. So one thing that came about with respect to your transplants is that it wasn't just your MHC was recognizing antigens. It was that your T cell receptor was recognizing MHC. So the restriction of T cell binding is to self-MHC. So how to think about this is if you have the same T cell, so here's T cell from a green person, and the T cells from the same green person, and the T cells from the same green person, and you had a macrophage from the same green person, and you had a macrophage from the same green person, and then you had a macrophage from a blue person. So the macrophage from the blue person would express blue MHC, and the T cell receptor from the green person, regardless if the peptide was recognizable, could not become activated. And then if you had a T cell receptor and an MHC molecule that were the same, there's from the green guy, and it had that same purple thing, then you would get activation. If you had the T cell receptor and the MHC from the same, but your peptide instead was a red peptide, the T cell did not recognize it, then you get no activation. So the T cell must recognize the MHC as self plus the peptide as foreign. And then both of those together is activation. Now, of course, sometimes the peptide is self and you get autoimmunity, but that's not quite what we're talking about here. Now, we have to talk about a few definitions with respect to MHC compatibility, self, non-self, and transplantation. If you are going to have a transplant and give it to yourself, this is called autologous. And that means self. And an example is skin. You can give yourself a skin graft, for example. If you have a twin and they're identical, and you get a transplant from your twin, this is called a syngeneic. And this is going to be tolerated. If you're going to get a graft from a non-twin, even if it is a sibling, this is allogeneic. And a sibling or general population is the same in transplant immunology and transplant rejection. And so really what MHC is, the major histocompatibility complex, is saying you can recognize self-MHC. If you give yourself a graft, you're okay. If you're an identical twin, it's the best outcome possible is a syngeneic graft. If you go out to the next sibling, you basically become allogeneic because your MHC loci is different from your siblings' loci. Now, the last thing I want to do is I want to talk about HLA subtype and disease. And so here we're going to talk about a few diseases. Ankylosing spondylitis is the first. This is associated with HLA B7, or B27. The key here, though, is it's class 1. It's that single B. It's not a 2-letter, it's a single letter. This is inflammation of the spine. And this causes a bamboo effect as you have fusion of the spine. Now, 90% that present with the disease have the beta 27, but only 2% beta 27 positives have disease. So it's associated, but not necessarily causative. Good pastures is another. And this is where you have antibodies that target the basement membrane of the lung and kidneys. And the basement membrane is, of course, the scaffolding structure. And so it holds everything together. It allows things to interact with each other. And if you have a patient who has bleeding in the lungs and also has potential kidney failure, you might start thinking good pastures, whereas if you only had one or the other, you would not think about good pastures. Now this is an HLA DR2, so this is class 2. And so this is CD8 mediated, and this is CD4. You have multiple sclerosis, which is the disease of the brain and spinal cord. And this has HLA-DR2 as well, so this is class 2, or CD4 mediated. And this is an immune system attacks the myelin sheath that coats your neurons, so you cannot send, send any signals. And then we also have diabetes mellitus. Which is also known as type 1. And this is where you have HLA-DR2 again. So this is class 2, CD4. And your immune system destroys your beta cells of your pancreas. So this is beta. And of course, the beta cells are insulin-producing cells. And so you can't produce insulin. So that's why it's called insulin-dependent. And then rheumatoid arthritis, or RA, where you get joint pain and damage. And this is HLA-DR4, which is class 2, CD4. And there's also systemic lupus. You get a butterfly rash on the cheeks. And this is because you have anti-DNA antibodies. And so when the UV light hits your face, it creates breaks in the DNA, and your antibodies are able to bind to that and cause inflammation. And so that's why you get this butterfly rash. And this is associated with HLA-DR3, which is class 2 and CD4. So I hope what you recognize here is that we have a lot of class 2 association with autoimmunity and very little class 1 association with autoimmunity. And that's because class 2 is going to be associated more with the antibody response, whereas class 1 has no influence on antibody responses at all. Okay, so that is the end of our